CD30 and cancer. CD30 is a transmembrane glycoprotein receptor that is normally expressed on the surface of activated T and B lymphocytes. Immunohistochemical analysis has revealed that CD30 is also expressed on the cell surface in some malignant cell types, including Hodgkin lymphoma, and some non-Hodgkin lymphomas, including systemic anaplastic large cell lymphoma. CD30 is therefore an appropriate target for an antibody therapeutic approach. Antibodies to CD30 have been used to target malignant cells, but with varying degrees of clinical activity. To enhance the therapeutic potential of antibodies, cytotoxic drugs have been linked to antibodies to form antibody-drug conjugates, offering the potential for targeted cytotoxic delivery to the tumor cell. Antibody-drug conjugate technology. In recent years, significant progress has been made in the development of antibody-drug conjugates, or ADCs. This technology has been used to develop several different agents for the clinical treatment of cancer. ADCs are composed of three parts: a monoclonal antibody conjugated to a cytotoxic agent via a cleavable linker. The key parameters in ADC development include the choice of target antigen, the potency of the agent, and the stability of the linker in circulation, and the release of the cytotoxic agent within the target cell. Peptide-based linkers may present advantages over earlier technologies, such as hydrazone and disulfide linkers. Peptide-based linkers are conditionally stable. They are cleaved after entry into the cell, providing targeted delivery of the cytotoxic agent. Adcetris, brentuximab vedotin. Mechanism of action. Advancements in ADC technology have led to the development of Adcetris, an agent targeting CD30 that is indicated for the treatment of adult patients with relapsed or refractory CD30-positive Hodgkin lymphoma, following autologous stem cell transplant or following at least 2 prior therapies when autologous stem cell transplantation or multi-agent chemotherapy is not a treatment option. Adcetris is also indicated for the treatment of adult patients with relapsed or refractory systemic anaplastic large cell lymphoma. Adcetris consists of 3 components: the antibody CA-C10, specific for human CD30, conjugated to the microtubule disrupting agent monomethyl auristatin E, MMAE, by a protease cleavable linker that covalently attaches MMAE to CA-C10. This linker is stable in systemic circulation, which may help to ensure that the agent is not released until it is internalized in targeted CD30-positive cells. Non-clinical data suggest that the biological activity of Adcetris results from a multi-step process. First, Adcetris binds to CD30 on the cell surface. Second, Adcetris is then internalized and trafficked to the lysosomal compartment. Third, lysosomal proteolytic enzymes cleave the linker and release the active agent MMAE into the cell. Fourth, once released into the cell, MMAE binds to tubulin, disrupting the microtubule network. In the final step, this cascade of events induces cell cycle arrest in mitosis, resulting in apoptotic death of the tumor cell. Adcetris, brentuximab vedotin, is indicated for the treatment of adult patients with relapsed or refractory CD30-positive Hodgkin lymphoma, following autologous stem cell transplant or following at least 2 prior therapies when autologous stem cell transplantation or multi-agent chemotherapy is not a treatment option. Adcetris is also indicated for the treatment of adult patients with relapsed or refractory systemic anaplastic large cell lymphoma.